Get started.
So it's like almost everybody, if not everybody's back. So we'll go ahead and get started. I think this one probably doesn't need any introduction, but he's going to be telling us about more fish and also contrast duration and atomistic modeling.
More of my nautical nonsense.
Yes.
All right, and so what I want to do in this next hour is tell you about, introduce you to contrast matching and applications and some specific studies that I've been involved in the years, and also a little bit about atomistic modeling, which is a very different approach to model-dependent analyses that kind of abandons use of shape reconstruction, all right, and directly reconciles an atomic model of some variety with its solution scattered directly. So, just to quickly review the fish, because there's a concepts in here that are important again here, right? So, intrinsic flexibility and disorder, all right, are innate properties of many of the open breathing frames in the eukaryotic genomes, as well as bacterial genomes, all right? The reality that crystallographers don't like to talk about is most of the things they crystallize can resolve and see the electron density of them, right? Aren't intrinsically disordered and floppy, right? Those are the things lost due to averaging. In a way, they wash away the electron density. All right, but a lot of biology relies on this intrinsic flexibility and disorder as normal innate properties of these macromolecules. Right, and so small angle scattering is wonderfully well positioned to directly address those properties, whereas other techniques may not be. Right. And so I'll just leave it simple like that. And so just to review, you know, with clamshells. These types of particles are really, really well suited for the ab initio shape reconstructions. All right, it's very straightforward, generally, to reconcile an atomic model of something that's very rigid with its shape reconstruction. Generally, there's not a lot of ambiguity in shape and so forth. And generally, it works really well. But again, with things that are heterogeneous in the different ways that I mentioned before, right, I'm not going to spend so much time here, whether it be heterogeneity like this, or heterogeneity like this. All right, we have to rely on other methods to get at structural explanations and approaches to our structural hypothesis. Okay, and so this we'll come back to with the atomistic model. All right, so I'm going to use you the first research problem here. So I spend most of my time researching HIV integrase, all right, which is one of only 3 enzymes in the retroviral genome. All right. And a long time, quite literally 30-year pursuit in this community is getting crystal structures in the full-length intact proteins. Right? So it's eluded such analyses to date where you can see all 3 domains clearly resolved simply because this protein is the devil's protein. It just wants to aggregate. You need one salt, glycerol, chaps, keep in solution. And so many people have turned to other retroviral integrases that are a little bit more distantly related to the human virus, right, to try to gain some insights into the chemistry it catalyzes, all right? And so back here in 2010, our collaborator, Peter Sharapanov, his group in the UK, made some landmark discovery. They found that a more distantly related prototype fomivirus integrase, right, could be crystallized with the DNA. So what integrase does, it 4 of them come together, and they grab the viral DNA ends, right, from the cDNA copy of the viral genome and incorporate it into the host chromosome, right? That incorporation event that happens very precise spacing, creates the provirus. At that point, infection is done, game's over, okay? That's the part that creates a permanent provirus in the host chromosome, and that goes on to continue the viral replication cycle, all right? So drug design is very focused on stopping that step, right, in the incorporation of the viral DNA into the host genome. All right. Peter's group has some landmark crystal structures of the prototype fomivirus integrase, right, as it was arranged around viral DNA ends. All right. Here are just minimal DNAs, like it's kind of hard to see from this angle. I'll show you more pictures. About 15 base pairs. All right. 2 viral DNA ends with a tetramer of protein. Right. We saw these structures, and Peter saw that he had some issues here, and that some of the domains were not resolved. Right. So he asked us to use our solution biophysics techniques to try to see if we can help bridge that gap. All right. And so the questions we had off the bat were, integrated as a 3-domain protein, but only 2 of the 4 monomers had all 3 domains visualized in the crystal lattice. I'll talk more about that in a second, right? And so we wanted to ask the question, how well is that intosome, right, which only with half of this is in the asymmetric unit crystal structure, how well is that derived intosome correlate with the solution properties? Is it really the bona fide tetrameric intosome, or is it just a crystallographic artifact? All right. And where are those missing domains? Kind of denoted here with these little asterisks. Okay. And then the question about the protein alone. This protein's unusual among retrolyte integrases. Unlike the HIV one, which is highly oligomeric, this is just a monomer in solution. So we're asking questions about what the protein looks like before it assembles on DNA, what type of conformational changes have to occur. Okay. There's no crystal structure of this protein alone, but it's very floppy, I'll show you. Okay. And again, it's a close neighbor of HIV. All right. So this is the problem I call the tyranny of the lattice. More often than not, crystallographers love to look at crystallographic packing and to make inferences and say, that's the diamond, and that's the monomer, and that's the active unit. But we don't really know because the fish are in the sardine can. We don't know how the fish actually swim in the ocean. So this is where the solution scattering is very useful to see if this dimer in the asymmetric unit bound to DNA really is related to this tetramer that's apparent in the crystal lattice packing. Okay, so that was one of the questions we had. This is what the reality is of that fish in the sardine can that can is. Okay, so the first thing we did was we get the expression construct from Peter, made the protein before any synchrotron trip, anything like that. We just first characterized it extensively using the techniques that we have available to us. Sec-mols, right? So you can make the intosome using their method, right? And it always consistently gave you the species that was very consistent with a tetramer of protein with 2 DNA ends bound, a 4-to-2 complex. Okay, now it's very consistent. And the monomer, both in sec-mols here, all right, was very consistently monomeric, not only by sec-mols, but also by cementation equilibrium analysis and cementation velocity analysis. All right. I have, together with Sophia Kendrick at Y Technologies, we have an application note coming on sec-mols using something called a conjugate analysis, where you can take advantage of the multi-angle light scattering and solve a series of linear equations to derive the mass of conjugate systems like protein DNA complexes, lipid membrane protein. So we're using this test case here to demonstrate that algorithm. So we'll have that application. If you have a sec-mol, this might be worth looking at if you're looking at protein DNA complexes or other composite particles. All right, cementation velocity to a single peak species here, right? That has this AS species and this roughly 2-point-something S species. I'll come back to this at the tail end about checking our result and so forth. All right. Okay. And so what did I do? So someone asked a question for how many synchrotron trips did you take to solve this, right? And so in each of these stories, I'll make a mention of how much I actually scattered it, right? And so we have a home source, a pen, alongside, I'm frequently going to synchrotrons to make measurements. This is the protein I took along. I just scattered it everywhere I could. I mailed it into ALS. I took it here to CHESS. Took it to APS. I want to see it did neutron scattering at NIST, right? I just want to see, I was really kind of curious. If I had the same protein, how consistent between different sources? And the answer is it was extremely consistent, right, with best practices. And I can get the same experimental result profile at many different locations for the same protein. And across a range of concentrations, this thing is exclusively monomeric, all right? This was a question in the literature that some people were observing diners based on a little tailed peak on gel filtration. This thing is monomeric by 3 different measures, okay? At least in our hands, okay? So now, you do the X-ray scattering experiment, okay? You can many concentrations and see RG is consistent. You compare the shape distribution function to that of the monomer observed in the crystal structure as bound in DNA. And the first thing that was very apparent was that the P of R's are very different, and the RGs are measurably different, all right? So whatever is happening in that transformation from the DNA free form to DNA bound form involves some type of conformational change that changes the distribution of the intertopic vectors. See here. All right, so something is different. All right. So we wanted to ask the question, well, what is that? All right. And this is where I said, oh, you know what? And so I'll just point out here that there are a bajillion, okay, features in the primary data, in the P of R fitting, all the way through the shape reconstructions that give you clues that your particle is flexible in intrinsic disorder. All right. Not going to cover all of them here, but this is sort of a short list of things to look for in your data if you think you're dealing with a flexible system. Okay. And these features are well documented. I think on my next slide here, I have some citations here. You can sort of look and get some ideas. Paul Bonato out in Spain has many papers, including the EOM software, where he goes into this in detail. And there's a lot of great publications more recently as well. It's a little bit out of date that speak to this issue, but also the utility of SAXS to get at this issue. All right, very powerful technique in that regard. All right, so the first thing I did, of course, was, hey, I'll run this shape reconstruction, right? So I ran 10, and they weren't the NSDs, normalized spatial discrepancy figures, which kind of measure the agreement between two calculations at a given position, were very high, right? So you couldn't average them together. I tried 10, I tried 20, tried 100, right? And I kept getting disparate solutions every time. All had good chi-squares, you know, and basically at the end of the day, it wasn't a very compelling solution. And this is because it's intrinsic disorder. I showed you the Kratky plot or the Pro-Divide plot, all the signatures of a very floppy protein solution. Yeah. So we have to abandon this and try a different way of getting at the question of what are the conformers occurring in solution. All right. If you ask anyone in the SAXS or SANS community how to go about doing atomistic molecule, you get different kinds of answers. And everyone has a preferred software and preferred calculations and approaches to doing many different steps en route to doing the atomistic modeling. Whatever the software is you choose, the general scheme is usually the same in every case, right? It starts with, you have to see what your available inventory is, whether the available crystal structures, homology models, experimental structures, whatever it is, what's available, building in the rest, right, using either electrodynamics, using all atom simulations, or coarse grain approach to use beads, okay, and building a complete inventory. So there's many programs that can help you do that. And then what you do is you use something called conformational sampling, all right? The idea is that each model you have is a hypothesis. So why don't we just generate Avogadro's number of hypotheses and see which ones work, right? And that's maybe overstating Avogadro's number. But we're talking about 10,000, 20,000, 50,000, 100,000 candidate structures, sampling all of configurational space. Something that looks like this. My friends at NIST call it the Nike plot, because a lot of times it looks like a Nike swoosh on your sneaker when you plot it out. Which each of these little dots being a single candidate structure, right? So you computationally generate all of these models. This will be done on molecular dynamics, Monte Carlo approaches, a program called SASSI I tend to use a lot, along with many others, right? And you can kind of impose chemical common sense, right, if you're using all atoms, but they have to obey a Raman-Condren space, for example, or other force fields, right? Then you have that test set, right, of structures, right? And then for each of those structures, you back calculate and generate a theoretical profile for that structure, right, a calculated profile, right? So now you have a big test set of calculated profiles, right? Now, you have that calculated profile set. Now you can compare those profiles to your experimental data, right? And then use a metric, chi-square or whatever it may be, with many different ones, okay? And see which profile or combination of profiles best agree with your experimental data, right? So you're testing a hypothesis here, basically, in a very expansive way, right? This is not shape determination, right? This is testing hypotheses, basically, right? And so the solutions you arrive at are not exclusive of other solutions that may also fit the data, right? So that's why we generate many candidate structures with the hopes that we can get a sense of the breadth of the solutions that make sense, right? A lot of times you might see a very flat distribution here with very low chi-square. It means that there can be a range of structures that actually equally agree well, right? And the more flexible it is, the harder it is to find one single structure that agrees with the solution scattering data. The solution scattering data is a volume weighted fraction, a summation of all different conformers that are contributing to that scattering. Right, so it kind of makes sense to know that a single structure will agree with that data in many ways. Okay, but this is the general approach. You look for the best single solutions and the best ensemble solutions, right? Using different methods, statistical methods, to try to get at that. Yeah. So what we did here, we used a program called SASSI, which is now much more well developed than it was when I was using this several years ago. Which was initially started by Joseph Kurz at the NIST Center for Neutron Research. He wrote it, and that has been expanded to his web-based program that has a lot of computing power behind it, ridiculous amount of computing power, very powerful, all right? Unlike some programs that are constrained with regards to the number of units you can have, protein versus DNA, and the type of sampling you do, SASSI is very powerful in that it uses molecular dynamics conventions to allow you to do a lot of different types of complex systems, all right? Including carbohydrates if you want, lipids if you want, all right? Now it works with protein and DNA specifically with regards to conformational sampling. Protein and nucleic acid. And And so it's developed quite a bit since then, but I highly suggest this program if you're interested. It's a really rigorous approach to it. And it also helps you with neutrons as well in contrast. All right. And so what do you do? So that's what we did here. No. Did you do that? Didn't like it. I do this. Yeah, the movie's not properly lit. Oh wait, it is. Okay. So you see this protein dancing here. It seems kind of ridiculous, right? But what we basically did here is you have this crystal structure, and we have regions of predicted disorder. What we simply did was we told the program, sample all conformational space, allowing very specific regions to sample available raw mitochondrions. Right? We developed a test that I think initially, I think for the publication, we used 50,000 test models. Actually, I went out to like 200,000 and found that it was overly redundant. We were able to sample enough conformational space with just 50,000. Okay. So this is a model that was built in molecular dynamics, where it's relaxed to the body of water. All right. And then allowed to sample many different candidate structures. And then what we did for each of those is we ran, at the time, I think we were running Chrysol. I think now there's so many other programs available that we probably wouldn't use Chrysol because it's more rapid. The problem with bottleneck in this analysis is generating those profiles for each and every of the candidate structures. We generated all the profiles, and then we started doing the chi filtering, right, using Chrysol or FoxS, as well as other analyses to look at the ensemble using both SASE and a genetic algorithm from the civils group called MacCHESS, which basically takes weighted fractions of a minimal number of sets of structures to try to find a minimal solution to your scattering profile. I'll show you that in a second. Okay. I just mentioned here, we decided flexibility based on other available information. In this case, was disorder predictions. There are other sources of information you can use to inform your interpretation here. I'll point out if you have HT exchange data, limited proteolysis, or other information, this is a great time to bring in that auditory knowledge. It basically will inform you how much you sample in the solution, that sample in the calculations. All right. How many structures? All right. This is going to vary from project to project. All right. This is actually a problem in this area that we don't really know at what point you saturate conformational space. This is a density plot showing up the sphere that was basically accessed by the different conformational samples that we did in this calculation versus just the starting crystal structure of the solutions that were preferred. Okay. And so somebody has to put some thought into in terms of if your test set is sampling all of the possibilities enough, not only reaching out there, but reaching out there enough to have it properly represented in your population. Sometimes you might reach out only once, where some solutions are conformers or oversampled and overly represented in your test set. Right. This is also a stage where you can put in decoys, put in the BSA molecule or something, just to make sure that you're not finding anything that fits that's actually specific to your molecule. All right, so this was the results of our analysis. This is our Nike plot, all right, where basically you have 2,000 structures here, plotted by their agreement with the experimental data. What you find here is the single best solution here in cyan, right, at a chi-square of 2.7, occupies position right here. But if we run the MacCHESS algorithm, all right, which is looking for the minimal number of structures that together in a weighted way best describe the data, actually found 1, 2, 3, 4 structures. Okay. Is this the only 4 structures? No. I'm certain if I ran the MacCHESS program over and over, like I did, right, you would find slightly different variations on a theme. Okay. But the general idea is that you would have an ensemble like this. If you overlay them, you might have one outlier like that together did a better job of describing the experimental data. Right. So generally, this provides a comment that you're not looking at really dramatic conformational changes necessarily, but basically changes relative to the catalytic core domain here. Everything is plotted with basically aligned relative to one domain. Okay. This is one way you can treat this, right? There are many other variations on this with regards to atomistic model. This gives you an illustration of what you can do. Okay, how many structures in a minimal ensemble? This is where you can walk along and try to find, right, at what point do you break off and start getting good solutions, right? So here, 1, 2, 3, 4. We stopped at 4. You could have made an argument for 1 or 2 as well, right? But I think what we're finding visually with the profile, chi-square is not great in Spark, but this will give you a visual criterion as well across the entire scattering profile. We found that 4 was the most appealing in terms of visual concordance with the experimental data across the entire scattering angle. Right? So it's important not just look at chi-square, but also look at your actual data to see where it deviates. And please pipe in with questions. I know I'm kind of going through this rather quickly. All right, so that's the protein monomer. That's about it for all talk about the atomistic modeling. Everything here from here on out, I'm going to talk about is contrast variation. Okay. And so why do we need contrast variation if you're a structural biologist? And so the intosome is a good example here. So you can do the SAXS experiment on the intosome, which is that 4-2 complex of protein to DNA, right? You can get X-ray scattering data. You can do the shape reconstruction. But there's a problem, right? This is an intuitive problem, is that, as Richard showed you this morning, DNA scatters much more strongly than protein in X-rays, right? So my data is naturally skewed towards the DNA component. So doing any ab initio shape reconstructions from that data is artificially skewed towards the DNA. So again, this operation here, not necessarily very useful in that regard, unless you're doing relative comparisons between two variants of the same thing. All right, and so this wasn't exactly what I needed to kind of get at this problem at the time. Okay, and so we need to use contrast to our advantage here to get at this problem because we have a composite particle. And this is just an illustration I had. Well, that's the sound of me recording a screenshot. All right, sorry. I'm like, why is there sound there? But basically, it's me doing the conformational sampling on this protein DNA complex, and I could never find any conformers using this approach that really did a good job recapitulating the SAXS RG. Right? That might be one of my kids crying in the back. My daughter. Sorry about that. That's kind of funny. I never hear the sound of it. So contrast variation. Why do we use contrast variation? So how do you explain contrast variation, right? So I love this far side cartoon from Gary Larson. So when the monster came, Lola, like the peppered moth and the Arctic hare, remained motionless, undetected. Harold, however, of course, was immediately devoured by the monster. All right. Lola here was camouflaged, right? So basically, she was kind of subtracted away from the view of the monster, but Harold obviously was not. All right. That's basically contrast variation in a nutshell. What you're doing here is you're leveraging, just in general terms, what you're doing is you're changing the scattering power of one component relative to the other. It's really the net. Like when you do a scattering experiment, you're subtracting the solute buffer and subtracting away buffer to get net scattering of your solute. Right? In the same way, you can change the relative difference in scattering power between A and B. Right? It basically changes what the net observation is. Right? So here with neutrons, right, what we're doing is we're leveraging a very peculiar property of neutrons. With X-rays, they come in, they collide with a molecule, and they kind of scatter off the electron cloud. Right? Neutrons are entirely different. Right? Neutrons collide with nuclei of atoms. Right? They're accelerated to quasi-elastic speeds, and they have a wave-like behavior when that happens. All right. Instead of talking about 1 angstrom, wavelength, talking about 6 angstrom wavelength with a little wider spread. Okay, but they interact differentially with nuclei. Okay, so they're very compositionally sensitive. All right, that's not just it's very compositionally selective towards the nuclei. All right, of the atoms that are scattering off of. So in X-rays, you can see there's not much difference between H2O and D2O, and you can see the protein has a higher scattering length density versus water, right? And deuterated protein doesn't matter. You can see the DNA is stronger scatterer than protein, relatively speaking. But in neutrons, okay, deuteration, the change of the nuclei, right, makes all the difference in the world. Right now, you have a very robust signal relative to the hydrogenated form. All right. So in neutrons, you have concerns with both coherent and incoherent scattering. Incoherent scattering being sort of background scattering. Not much of an issue with synchrotron X-rays, but with neutrons, this is a big deal. All right. And so what we do in neutron scattering is we optimize our signal using deuterated components, whether it be deuterated water in the solvent, or deuterated protein, or deuterated lipid, or something that's deuterated. And we leverage that to get specific scattering power from that species as it exists as part of the larger whole. All right. So The other story I'm going to tell you about, basically themes and variations of the same stuff here. Different systems and different applications. Different biological questions. Yes?
So at one point you rotated, those extended portions. So you have, tags on the protein that you're doing SAXS on, but the crystal structure that you're basing it off of doesn't have those tags. Is there a way to artificially construct them and add them to your crystal structure?
Like missing inventory?
Sure.
Yeah, I use NAMD. So there's two programs I'll use routinely to do that. One is CNS. CNS Explorer has a function, NMR function that will build out missing inventory and idealize Raman-Condrins. I also use NAMD, the Molecular Dynamics program is free from the University of Illinois. That will also do the same thing. Or you can use some of the AdSAT software and build beads in that situation as well. I like the former, the atomistic approach, because then you're using Raman-Condrins as an additional filter for reality. Beads aren't obliged in the same way usually.
All right, so I think we have the sound back, so we can proceed.
All right. All right, let me give you another example. That group 2 intron again, okay? So, not everything can be done by neutrons, all right? And that can be because you don't have enough sample quantity, all right? You don't have access to neutrons, all right? Doing neutron scattering is difficult in practice. You have to be patient because, A, there are only 2 sources here in the U.S. that are really appropriate for biological SANS. That is Oak Ridge National Labs. They have 2 reactors there, and the NIST Center for Neutron Research in Gaithersburg, Maryland. At both all those beam lines, they're generally talking about beam lines that are oversubscribed 2 to 4 folds. It's very competitive to get beam time to do these experiments. They only have cycles maybe twice a year. All right, you have to be very patient. So sometimes you can't get access to neutrons right away in the way that you might need, or you don't have enough sample. Right, neutron experiments, you need a lot of sample. All right, so we're talking about 350 to 700 microliters in these QF banjos at a time to do the solution measurement. All right, sometimes it's not realistic in some systems that don't express well and so forth. Okay, so sometimes we want to use X-rays. So we can do contrast variation with X-rays and do it very well. All right, in this case here, if you're looking at a protein nucleic acid complex, though, the reality is you can use sucrose or glycerol to contrast the way the protein leaves behind the nucleic acid signal. Right, whereas with contrast variation with neutrons, there's a wider range where you can contrast either component away. Okay, and so here we have a problem where it's basically here this is an example where we're looking at this group 2 intron particle, right, which is a 440 kilodalton complex, 70% RNA, 30% protein. Okay, and so we wanted to know where the protein was located relative to the RNA component. Right, and like most things I do, I make a prediction and I'm wrong. Right, which I love because it's a general running punchline with me. All right, and so going to this, I was convinced that the protein is not going to be at the middle; it's going to be somewhere on the edge. It's going to bind at some edge. And I just thought, it makes sense. All right, and so you can simulate this data, and then with increasing amounts of sucrose, you can make predictions of what these germinal plots in this window would look like using X-rays. All these experiments that I'm going to talk about now, we're done here at MacCHESS. Okay. With very small amounts of sample. All right. They at the time when we were doing experiments, they had a customized 15 microliter sample cell. Right. So we could use a minimal amount of material to do the X-ray experiment on. Right, which was ideal for a situation where I only had 10 micrograms of this stuff. Right. I was leveraging the fact that big things scatter very strongly at small concentrations. Right. If you're doing X-ray scattering on a ribosome, you can work at 0.2 mg per mil. Get really strong scatter. We're working on a nucleosome. You can work at 0.75 mg per mil on a mononucleosome and get very strong scattering. It's 200 kilodaltons and a lot of nucleic acid. Right? Group 2 intron, 7% RNA, 440 kilodaltons. It's like work down at the low, mg per mil regime and even lower and get very strong scattering. Not the case of working from that 8 kilodaltons, for example. Exact opposite. Right? All right. So this is an experiment I did here at MacCHESS. It was not elegant in that, these tiny amounts of samples and we're exchanging in increasing amounts of glycerol up to 65%, and the data quality drops off very precipitously, and that the intensity and the amount of Q was dropping off. But it was enough, the variation in I0 and RG was enough that I could put it into MANSA, right, and derive a 2-phase model, right? At the time, there was no experimental structure of the group 2 intron. nothing. All right, even the homology models were garbage. All right, and so all I could do is really make comment about the disposition of the parts relative to the whole. All right, and so here's a monolithic solution, right? And so based on this, we made some suppositions about which of these domains were based on connectivity. All right, and there's a paper citation. You don't have to take pictures of it. There's a citation. You can find the paper and you click acid research. Right, and so that was a prediction in 2010. Right, Marlene's group was also concurrently working on a high-resolution EM structure, which came out a few years later, and we weren't really far off. Right, this is a little side note. If you're doing electron microscopy, small scattering can be a very useful tool to validate and check your class averages and your reconstructions. So during the course of the initial phases of this project, we took their initial negative stain envelopes and checked it against the scattering data. Back calculated profile and checked it, and we told them that they were on the right track because they agreed with the solution scattering very well. Right? They were very excited about that. So we kind of used that as a quality check, and it turns out that we were pretty close in terms of placing the protein towards the middle. Obviously, this is a much higher resolution than the smallest scattering, but the smallest scattering was generally consistent. So it was kind of gratifying that in that particular case it worked out pretty well with a very modest amount of material. Right? The nice thing about the smallest scattering technique, it's versatile. If you have a problem, you probably can find a solution to it working at the sample environment stage. Okay. All right, third example. All right, it's not even a protein. This is great. So what are reverse micelles? Reverse micelles, all right, are a rather interesting situation where you have basically an organic solvent, right? You have some detergent like CTAB, right? And then you have an aqueous core. So it's kind of turned inside out from what you would think you're accustomed to. And the idea is that you can encapsulate things in the aqueous core, right? Whether it be for program release or for, in the case of John Schwann's group and Kim Sharp at Penn, with applications to NMR, right? Improving NMR spectroscopy, all right? Or other high concentration measurements. So the reverse micelle technology has a large number of applications potentially in pharmaceutics and in structural biology and so forth. All right. However, in order to make those applications I just mentioned, you have to have a very good understanding of the nature of the micelle being created, right? And structural information is not readily accessible by most techniques, right? And so what Kim did was he developed a very robust multidynamic simulation, 1-2 nanoseconds, of these molecules, assembling them basically from a distended state down into a assembled micelle, and then benchmarked the simulations using small angle scattering. How many trips? This was 15 minutes. 15 minutes at Oak Ridge National Labs, and this was an afternoon at a home source at Penn. All right, it was a well-planned experiment in the right samples. Okay, and so what you do is simultaneously fit all three profiles and then derive a 4-shell model, right? And determine the width of this shell, the width of the core, right? And check it against your MD simulation. This is a very common application of small angle scattering, is to experimentally benchmark multidynamic simulation, a reality check, and kind of tune your force fields and just check your simulations. This is a very common application, especially in lipids literature and reverse micelles and colloids. Okay, in this case here, we're able to nail down from the small angle scattering versus simulation the size of the outer leaflet in the CTAB, the little bromine shell that occurs in the inside from the salt of CTAB, and the aqueous core. Next step now is see if we can encapsulate proteins in there and do the same thing with proteins in the reverse micelle. Okay. Kind of geeky, but it's a lot of fun. All right. Fourth example, the final example I'm going to tell you about, all right, the last remaining bits of time, is revisiting that nucleosome. All right, so this is an experiment that was done back in the mid-70s, before Brown Tunnel was born, basically, date myself. All right, and we're kind of revisiting a classical experiment, or a canonical experiment, with an application towards variant nucleosomes. Right. And so I collaborate with Ben Black's group, just one more the Penn, where they're very interested in the centromere. That is these little nubs here at the middle of the chromosome. All right. What's neat about the nucleosomes at that position there is they're functionally different, in part because they wrap around very different types of DNA. They're called alpha satellite DNA, which is greatly enriched in A's and T's. Right. Additionally, they have an alternative histone. Instead of having an H3, they have something called CENT-A. Right. The combination of the different DNA and the different histones confer different biological properties at the centromere, but not found in other parts of the chromosome. Okay? This is work we're writing up right now for publication. And so what we wanted to do was we wanted to find out why it is that they were different. Right? If you just take all 80 or so nucleosome structures in the protein data bank, just superpose them, we find it two fundamental problems. Right? All those nucleosome structures are within an RMSD deviation of 1. All right? They all pack in very similar ways, like as if they're in chromatin. And secondly, they're missing a ton of atomic inventory. Right? 30% of the protein inventory of a nucleosome. So protein is 50% of the nucleosome. Right? Another 50% is DNA. 30% of that protein mass is entirely unresolved in protein crystallography. Right? The business end of things, the histone tails, the N-termini, the 30 or so amino acids on each of the 8 histones in an histone oxide. Right? Don't have any information on that stuff from other techniques, other direct sort of techniques. But here, you can sort of ask questions and account for that inventory in our analysis. Okay? So we want to ask a question: if you start mix and match different histones, different DNAs, right? What combinations gave you? Which combinations, if any, were different in solution, and how they compare with their crystal structures? All right, and you can go about this couple, and we tried this both ways. All right, you can do the X-ray experiment where you contrast with, in our case, sucrose. Where you contrast with the protein component. Okay, that's one way of doing it. And in practice, right, you're kind of limited in the range of the scatter length density you can probe. Okay, and you're only contrasting with the protein. All right, and you can also do neutron contrast variation. All right, where you do the isotopic exchange, not by deuterating the protein in this case, but what we did was we simply changed the percentage D2O in the buffers. Okay, this has been, so I'm not going to bother laboring that, other than you can just calculate the scatter length densities for each composition of different nucleosomes, and it's slightly different in every case. All right, this has been a project several years running now. We've characterized 6 different variant nucleosomes. All right, and we've made extensive use of synchrotron X-rays, home source X-rays, and 2, actually this is missing NIST here too, 2 different neutron sources, right? And bringing all this information together to try to gain these insights. So this is something that's been a long time coming. Going back to your question, how many trips? Too many. I don't even want to talk about it. Okay. All right. And so first thing, the question we want to ask is, can we take the crystal structure of a nucleosome, right? Build in the missing parts, and then reconcile it with the solution data. All right. The most basic of experiments. Can we take a canonical nucleosome, reconcile it with a solution scattering and its biophysical properties? And the short answer is, we think so. Yes. All right. So here are molecular dynamics built models of canonical nucleosomes, both for the classic H3H4 set octamer and the Sempé derived. Those blue blobs are basically beads, or basically representative beads. They're actually all atom models of those missing histone tails. And the MD simulation, in a box of water, they come down, they kind of collapse on the DNA, which kind of makes sense with a charge, DNA's charge, so they want to get together. Okay. If you back calculate from these models what the cementation coefficient is and the frictional coefficient, you get very good agreement with the experimentally determined S values. Right, 12S, 11 to 12S is the classical number. The literature going back decades. Right, we get generally consistent results in both cases. So it's good agreement on that level. All right. As mentioned before, it's not trivial to back calculate from in X-rays from a crystal structure to a experimental profile if you have a protein DNA complex. And that's because the water boundary around a protein DNA complex is very different and can't be approximated like you can for protein alone or DNA alone. All right. So in practice, I think with the newest servers, I'm using the MacCHESS server here from a group in Germany, which is very robust, where it does an explicit solvent calculation to help you do this calculation. All right. If you do the explicit solid boundary calculation, don't use any approximations, right? And then check it against SAXS-WAX data, right? You can get very good correlations between the canonical model and the solution scattering experiment. Okay, and so we were happy with that in that regard. All right, although there's some aspects of X-rays that are a little bit misleading in terms of, I'll get to that in a minute, right? How about the neutron experiment, right? Here are the experimental Sturm plots for the neutrons. So the original Drexel paper in 1977 plotted a straight line, right? And I was first concerned about, oh my gosh, why is this parabolic? What did I do wrong? And so I presented this in NIST a couple of years ago, and the old guard, I totally regarded in the most favorable way possible. The people who originally did these experiments, right, were working on the reactor kind of said, that's fantastic. I'm like, why? Because back then, they couldn't reliably get some of these wane points, these contrast points, because they were dominated by incoherent scattering. The technology has advanced so much over the course of 4 decades that now you can, with the available flux and detector technology, get much better determinations of these contrast points at these harder to get places, right, the wane points. And so this parabolic nature is for real, right? And it agrees pretty well with the experimental models. So you can see you can back calculate a model, get a theoretical Sturm and see how well it agrees with the experimental, both the classical histone case and the Sempé-derived model. But we can do that. We can take a model of a nucleosome and basically reconcile it with the solution properties in the most rigorous way we can, which is a reasonable starting point. Now, if we want to ask what differences may occur later. All right. All right. I mentioned before, if you don't have the right inventory, you can't make any meaningful calculations and comparisons. All right. This is a good example. If you just take a crystal structure, which has no tails in there, and try to compare it and do the theoretical Sturm, you can see that it's not even close. Right. Only when you have the extended tails built it in, right, get closer. Then you find with the MD simulation, when it's kind of pulled down, you get a good correlation with the experimental data, right, as I showed you in the previous slide. But having the missing inventory makes all the difference in the world, all right? That should precede any rigid body modeling and any atomistic modeling, right? You have to have the inventory first and then do the rest of the model. Don't do it the other way around and make mislead yourself. Okay. All right, now, how about the Sempé-derived nucleosomes? Now here, what I call 602, these are the alpha satellite-rich DNAs, okay? First off, we just take these particles and assemble them, and we do in this experiment, we reconstitute, which is very laborious, run it again, reconstitute a separate batch, do it again. Did that several times because we were calculating a different number, all right? I mentioned before in the talk this morning, when cementation coefficient goes down in stoichiometry, they say that means the frictional coefficient is going up, all right? So these particles, by cementation equilibrium analysis, were octameric nucleosomes, right? But they have a much lower S value, implying a more elongated shape in solution, right? There are crystal structures of Sempé nucleosomes around DNA that almost perfectly coincide with the canonical models in the crystal labs. But here in solution, seeing something different from cementation velocity, right? Sec moles and cementation equilibrium show that these are octameric nucleosomes, our preparations going into neutrals, right? Now, how about contrast variation? So this red and blue here, what I showed you in the previous slide for the canonical, the histone octamers wrapped around the tight 601 DNA, form very nice, discrete nucleosomes. In comparison, contrast variation profiles for the ones wrapped around the alpha satellite DNA are very, very different, kind of like flat lines, right? The conformation is very different in solution. And what we're doing right now is trying to figure out structural models that may explain this behavior in solution, both with regards to S value and the Sturm profiles, right? Mentioned this morning how important concentration determination is, right? This was a major roadblock for a long while. We had to go back through all these notebooks for several years, right? Because we were using one form of concentration determination, which turned out to be 30% off in concentration, which you might get away with just simple structural modeling. But the nice thing about contrast variation, that I is 0 term, that it's exquisitely compositionally sensitive. You should be able to back calculate from all this neutron data the precise molecular mass here, corroborating molecular mass for your particles. And we can do that. So here, our collaborator Susan Kruger wrote just a simple algebraic program that basically takes all the data and solves a system linear equation to derive molecular mass for a two-component system, protein and DNA, or in a case where you fix the DNA and just calculate the protein. And all around, you get figures now with the right concentrations. Angry about this. You get concentrations, masses that are now very consistent with an octameric nucleosome, corroborating the other experimental measures that we made. All right. At first, we thought, oh my gosh, these numbers don't make any sense. When we went back in the notebooks and looked at our Bradfords versus our nanodrops, we found that one was 30% off from the other. We had to do a correction to account for that deviation. Right? So be very careful. Again, concentration, especially with neutron scattering, you have to get it right. Otherwise, you're going to lose out on a major part of these analyses with regards to deriving mass and molecular volume. Okay? And so that was gratifying to figure that out. That's why notebooks are so important. Right? All right, and so in short, we were able to determine all these figures of many different nucleosomes, all right, and we're working on publishing this. But this kind of illustrates revisiting this canonical measurement with modern technology and approaches is kind of fun, because you kind of just discovered how much technology has advanced and allowed you to extend the knowledge in this system that had been worked out many, many decades ago. Okay. Kind of worked out pretty nicely there. All right. Contrast variation. All right. Papers on SAXS using macromolecules come out at a clip of about 1 to 2 per day in PubMed. Right. Whereas in neutron scattering, papers come out at a clip of about 1 to 2 per week. Right, on macromolecules. There's a reason for that, but contrast variation is very powerful. I highly encourage you to make use of it, whether it be with X-rays or neutrons, because it can really enhance your study, and especially when we're working with composite systems that are more than one type of macromolecule. All right. All right. With that, I'll take any questions. We're on time. Questions?
I do.
Yes.
So, can you go back to the slide where you have the Sturman plots for the truncate out further?
Here?
More. This one, yeah.
Okay.
So you have your atomistic models there. Like if you took one of these sort of truncated models that don't have any of the tails, that was completely disordered, how would the IOQ and the Q plots look like?
It would start out, it wouldn't be that far off, I think, historically. So this is a very wide angle scattering for personal planets that goes up to 1, right? Generally, you do pretty well, but here, there's a little characteristic inflection in the literature. And you see it too, just angle, you can see it practically with pure mononucleosomes right here. Getting a good fit in this peak feature here, and then these water properties out here, these higher Q, it becomes more discrepant. Right? You can't get a fit in this lower Q regime, but these higher Q features are more difficult to fit.
So in other words, you didn't do any of this contrast stuff. You wouldn't be able to resolve
This is what I love about this study. If you just did X-rays alone, you can convince yourself that every nucleosome I looked at was exactly the same.
That's right. That's what I was getting at.
This is my favorite part of the study. It's a very obscure, very, very esoteric point, but it's such an important one. Because of contrast, right? People can scatter nucleosomes out the wazoo and convince themselves that all of them are identical, statistically identical. But I think that's phenomenology, because the X-ray scattering point is right here. And you put all of these Sturman plots together and overlay them. They all cross right here at the X-ray point. But at these different contrast points, they all vary, right? It's the contrast variation that's revealing the differences, right? Which are otherwise not resolvable by the X-rays alone. Right. I just showed you the originally I scattered this by X-rays, but oh my god, these are all the same. Oh my gosh! But when I did the neutron experiment, that's when I'm like, moly, these are really different. Okay, it's because it's just this funny situation where all the Sturman profiles for all these varying nucleosomes all cross at the same point, at the same contrast point, which happens to be X-ray. Right. It's a math problem because not all protein DNA complexes have the same proportion of protein and nucleic acid. It's going to be different for different systems. Right. But in this case here, it's just like, oh, X-rays are in the wrong place. If they were just over here or over here, I would have seen these differences. So 70% D2O and 20% D2O is the biggest difference in neutrons. Pretty good. Okay.
So I think, Tom, you're up next. So next we're going to hear again from Tom, and he's going to tell us about a very exciting new method that just got published, 2 months ago, give or take, calculating actual electron densities from SAXS profiles, which, as Richard was saying this morning, previously we haven't been able to do. So this is something we're very enthusiastic about, and something you'll have a chance to play around with tomorrow in the tutorial. So.
All right, so as it was alluded to earlier, I'm going to talk about a method I came up with to calculate the electron density function from solution scattering, which effectively means that this is solving the phase problem for solution scattering. If you're familiar with crystallography, a big part of crystallography and why it has been successful over the last several decades is that people have figured out ways to solve the phase problem. So here I've done that for solution scattering. Okay, skip. All right, so pretty much all of the ways that we get information, three-dimensional structural information from solution scattering is through modeling, okay? And so this is developed historically over the last few decades in different ways. So it was first by doing simple shapes. So effectively what you do is you say, I know I can create, ellipsoids of different axial ratios. I can create rods. I can create, donuts or core shells or whatever. And I can use that to define an envelope. And inside of that envelope, I'm going to assume uniform density. And so by doing that, then you can fit these different ratios of, the dimensions of your model. And that can give you some really, really basic low resolution shape information. And then there was the development of including multiple different types of shapes using some Monte Carlo approaches that allowed us to get a little bit more complex. And then a big advance happened when Sturgeon came up with the idea of using spherical harmonics to define an envelope. And so this, rather than using simple shapes, you can actually get much more complex shapes using spherical harmonics. So rather than using spherical harmonics to define the density, what it does is it uses spherical harmonics to define a distance from the center, and so that defines, in 3 dimensions, and so that defines this envelope. And so we assume uniform density inside of that envelope, and again, we can calculate the scattering profile and iterate through lots of different possible spherical harmonics coefficients and come up with something that roughly matches our data. But this has some problems because you can't create particularly complex models. You can't have things with cavities or really wild shapes. So really what you see nowadays is bead modeling, and this was a big step because this is a way of creating very complex shapes using these sort of coarse-grained beads, and you can do this iteratively by assuming all of the beads have the same density and then calculating what the scattering profile is from millions of different iterations and coming up with a model that best matches your data. And then over the last 2 decades, by now, there's really been a huge advance in terms of hybrid modeling where you can combine all these different pieces of information together. So I would sort of put all under this all under the heading of modeling, meaning you're generating lots of different models, calculating scattering profiles, and then seeing which one best fits your data. So what I'm going to talk about is how we can directly calculate the actual electron density function from our scattering profile. Okay, so I alluded to this earlier, but this is, it's going to go, oh, there it goes. This is a 3-dimensional representation of reciprocal space, okay? So this up here is that image I showed earlier, and this is a 2-dimensional cross-section, a slice of the Ewald sphere through this 3-dimensional reciprocal space, okay? So this 3-dimensional reciprocal space is unique to the structure of your biological molecule, okay? So if you took the electron density function and you calculated a Fourier transform, you would end up with this. What I'm showing here are actually the intensities, but in reality, the molecular transform structure factors is a complex function where it's got amplitudes and phases at every point in 3-dimensional reciprocal space, okay? So this is what reciprocal space looks like for your protein. Well, this is what reciprocal space looks like for lysozyme. For your protein, it looks something similar. Okay. So the problem that we have to solve, be it crystallography or single molecule imaging or whatever, is that we can take these intensities, which assume for a moment we could do single molecule imaging. What you would get at the end of your experiment is something like this. Okay, you get a 3-dimensional volume that would assign intensities to every point in reciprocal space. Okay. What you get from those intensities are the amplitudes, okay? So you have all the amplitudes of the structure factors, but what you don't have are the phases. And so what you need to do is solve the phase problem, okay? So we know our 3D amplitudes, but we have to solve the phase problem. All right, so if we just sort of keep ourselves in sort of single molecule imaging mode for the ideal case, we can get an idea of how they do this. And they do this using a process called iterative phase retrieval. And I say they do this, but single molecule imaging at this stage is not really a thing, sort of. The single molecule X-ray diffraction, at least. They've done some initial proof of concepts with giant, Mimi virus type particles with XFELs, and it is becoming more practical, but it's still a ways out. But this is how they do it. So what happens is you have your three-dimensional intensities, and therefore your three-dimensional amplitudes of your structure factors that you know. You don't know your 3D phases. And so what we can do is we can take advantage of what's called oversampling, the fact that the reciprocal space intensities in single molecule diffraction are continuous. It's not like Bragg sampling in crystallography where we only get these discrete peaks, these delta functions. In scattering, in single molecule scattering, you get these continuous fluctuations throughout reciprocal space. And so that oversamples this, Shannon sampling that I had mentioned earlier. So the way that we take advantage of this is by thinking about our electron density and structure factors in two domains. So we have our real space representation of our molecule, and we have our reciprocal space representation of our molecule. And these two spaces are in fact equivalent. They're just related by Fourier transforms, okay? And so what we do is we start with like a random electron density, okay? You take a volume of space that contains your particle and you just set it to random numbers. And then you calculate a Fourier transform, and that gives you a set of structure factors. So this is now a complex function, whereas the density is a real function. So there's just one number at each point in real space. Whereas in reciprocal space, there's two values at each point. There's the amplitude, which is the real part of the function. And then there's the phase, which is the imaginary part of the complex function. And so that gives us a set of random structure factors. But what we know from our single molecule scattering experiment is we know what the 3D amplitudes are from our 3D intensities. So we can make all of those random three-dimensional amplitudes, we can set them equal to what our experiment tells us, okay? And so that gives us a new set of amplitudes. With the original random phases, we have a set of new structure factors. We can calculate an inverse Fourier transform to take us back into real space. And this gives us a new set of electron densities from which we can now play with, okay? And we play with them by applying real space restraints. And so these are things like positivity. So positivity means you can't have negative electron density. You can't have a negative number of electrons in a given space, right? We also know that the solvent, as a rough approximation, is typically flat. Bulk solvent is flat. We also know that particles are connected objects, okay? You don't have disconnected particles. They're single connected objects. And so we can modify our electron density function with those real space restraints, get a new set of electron densities, and then we can iterate through this process. So that sort of gives us a new set of structure factors. And then we can modify those structure factors with our experimental amplitudes in 3 dimensions, keep our original phases, or these new modified phases, and do, again, an inverse Fourier transform. And we cycle through this process. And what happens, that's why it's called iterative phase retrieval, because we're iterating through this process. And as we iterate through this process, they get better and better and better until it converges and you have your structure. One of the key pieces is that this uses something we call a support. So a support is effectively the envelope of the particle. So this is how we say the solvent is where we flatten the solvent, and the particle is where we make sure there's positive density and that it's connected. So this is through the use of the support. And I'll explain how we do that. And then we iterate until we retrieve our phases. All right, so how about for the case of solution scattering? How do we apply this basic idea of iterative phase retrieval solution scattering? So again, we start with our molecular transform. Just imagine this in 3 dimensions. And as I mentioned earlier, because of spherical averaging, because the molecules are tumbling in solution, we end up with an isotropic scattering profile. Okay, so this is a 3-dimensional function where we're just, it's radially symmetric. And so we're taking this cross-section through this 3-dimensional function. And so what happens is that because it's this spherically averaged function, we don't have 3D intensities, whereas in the single molecule imaging case, we had a set of 3-dimensional intensities that we got from our experiment. In this case, we only have 1-dimensional intensities, the spherical average of the one-dimensional intensities. So rather than just having to solve the phase problem, we have to solve what I call the structure factor problem. So we have to figure out what both the amplitudes and the phases are in reciprocal space in order to figure out what our structure is. So how do we do that? Well, it's a very similar algorithm I created that I call iterative structure factor retrieval. So rather than just iterative phase retrieval, we're going to get our amplitudes too. Very similar though. So we start with random electron density. And to sort of make this as a sort of a computable thing, we can't have continuous numbers, or at least I'm not using continuous functions. I sort of discretize space to make it, good for computers. And so I just create a three-dimensional grid. It's just a cube with a bunch of cubes. Okay, we call them voxels. And so you kind of think of it as like a three-dimensional image. If you think of like a CT scan or an MRI scan, that's what these are. They're three-dimensional images. So we fill this with a random electron density by assigning each one of these grid points a random number. If you think of like an image, when you look at a 2D image, it's just a bunch of pixels that each has an intensity value, right? Grayscaling. So we take that random electron density, we calculate the forward Fourier transform, and that gives us a set of structure factors. And by nature of the Fourier transform, we have a grid that's exactly the same size. So if this is n by n by n, we end up with an n by n by n Fourier transform. So we can, from that set of structure factors, the set of structure factors again is a complex value. So each grid point now has a real value and an imaginary value of the complex number. But we can calculate a set of intensities from that set of structure factors, which the intensities are just the structure factors times their complex conjugate. So this is all the same as what we do for single molecule imaging. So what's different about solution scattering? Well, remember that what we have is a restraint, is our experimental data tells us what the spherical average of the intensities are. So what we do is we bin all of these intensities into concentric spherical shells. And so what we know is that the spherical average of all these grid points within that shell should equal our experimental data. So we calculate what our calculated spherical average is, and we can compare it with our experimental data. And what we know is that each one of these spherical averages of each of these concentric shells should equal that value right there. So what we need to do is make those intensities equal that value. So the way that we do that is we scale the three-dimensional structure factors of each of the grid points all by the same number. So that's the key. We're going to scale all of the intensities within that concentric shell by the square root of the ratio of these intensities. Since this is intensity space and we're looking to scale the amplitudes, we do it by the square root of the ratio. And so what that does is it scales this data point up to our experimental value. And so that scales all of these grid points by the same number. And so now we end up with a set of intensities, where we end up with a set of structure factors whose corresponding intensities match our experimental data. Spherical average. Does that make sense? All right. So that's the key piece. So then everything else is the same as single molecule imaging. We calculate a inverse Fourier transform from this new set of structure factors, and we end up with this very noisy and very poor initial electron density map. And the reason for that is because we started with random density, so we have random phases. And when we're scaling all the intensities, or all the amplitudes here, we're not doing anything to the phases. So we have two problems. One, we have a set of random phases, and two, even though we have the average of the intensities being correct, we don't know that the actual intensities at each grid point are correct, right? They're likely very wrong, because we started with random numbers. So this is where the real space restraints come in handy. It turns out that you can apply this same iterative cycle of applying real space restraints to not only recover the phases, but it also recovers the amplitudes, just by using these restraints. All right, so we have this sort of noisy electron density map. And as I mentioned, we need to define the particle. So we define the support, which we use a process called shrink-wrap for this, which effectively looks at where's most of the density. It just blurs the noisy map and makes it a less noisy map. And then just says, where's most of the density? And it says, okay, let's define that as the boundary. And we're going to set all this noisy stuff out here to be 0. We're going to flatten it. And then we're going to set all the real density inside of it. We're going to make sure everything's positive. We're going to make sure nothing's negative. And then that gives us a new sort of flattened, cleaned electron density. But now that we've changed the electron density, the corresponding experimental comparison to the experimental data no longer matches. So we have to calculate a new set of structure factors and a new set of intensities, calculate the spherical average again, and do this process over and over again iteratively until we achieve convergence. And so that's the basic idea. All right, so here is a little animation showing this process. Here's our initial grid. It starts with random electron densities, just uniformly distributed random values. On the bottom left here, we have the scattering profile fit. So we have in black dots the experimental data. We have, rather than using the experimental data, we actually use a fit to the data. So for example, the program that runs all this is called DENSE, and you'll see this in the tutorial tomorrow. And so to run it, you can actually run genome, which it accepts genome, thanks to Jesse, accepts genome as input, genome output files as input to the DENSE algorithm. So it actually is using the smooth black fit to the data, and that takes advantage of the oversampling, effectively increasing our signal noise. And so you can see in the red dots here what the current spherically average scattering profile from this random grid of density is. And you notice it's completely different than what we would expect, right? And that's not to be unexpected. It's random. It shouldn't look anything like a particle. And on the right here, we have the measure of the fit between the red dots and the black line. All right, so as we start the animation, you notice that it goes from this random thing to this blob virtually within the first step. And the reason for that is because once you take all those red grid points and you scale it to something that looks like this scattering profile, it looks like a particle in reciprocal space. So that's why it sort of gets globular. And you notice that it decays. What happens with these spikes is that every once in a while we update the envelope. As it gets better and better, we do the shrink wrap algorithm again, and it gets better. And you notice that over time, it converges. And this process does allow it to get out of these local minima through this randomization. And you can see that it's a low resolution electron density map. Again, this is BioSAXS. Everything is still low resolution. But if you show what the actual structure is, you can see that the fit is pretty decent. Now, as I mentioned, each time we run this, we're running with a set of random density. So every time you run it, you get a different answer. Just like in bead modeling, if you run it 10 times, you get 10 different answers. So we can do averaging just like we do in bead modeling. Okay? So this is just one example of something that sort of looks like a dumbbell with some spherical shells on either end. And so we can calculate this 20 or 100 times, something like that. And we split it into two separate, oh wait, not splitting it yet. For averaging, we can just align all these and end up averaging what the density looks like. All right. Now, we can actually, using this method, get an estimate of resolution. And so we're following, in this case, the basic idea of estimating resolution the same way they do it in cryo-electron microscopy. And so what we do is we take our set of 20 initial random configurations, or convergence, the results of these initial random starts, and we split it into two sets, just randomly, even and odd, for example. And we calculate two independent averages, okay? Then what we can do is we can actually compare these two. And that gives us a comparison of this sort of even versus odd, this black curve here, the black solid line. And from that, we can actually look in reciprocal space. This is a Fourier shell correlation. We can look as a Fourier shell correlation, what resolution, so you can see here, it's very well correlated at very low resolution. Resolution is in inverse angstroms, 1 over angstrom. So at 0.01, that's a resolution of 100 angstroms, okay? You can see that they're very well correlated, and just as a general rule of thumb, people like to use the idea of where, defining the resolution where the Fourier shell correlation falls below 0.5. And so that says that in this case, the resolution is about 26 angstroms, okay? And so this is actually a way of estimating resolution from our BioSAXS reconstructions. So one question is, this tells us how sort of internally self-consistent these different reconstructions are. But one question is, does this actually tell us how well it matches the actual structure? So what we can do is we can do some simulations. And that's what actually this is. So we can actually calculate SAXS data from a simulation and compare the resulting Fourier shell correlation of the two halves to how well does our average match our structure. We can calculate a Fourier shell correlation from that. And it turns out that for the most part, that's what the dashed line is. So that's the known structure versus our actual envelope. You can see that, while not perfect, it generally, we did this for like 12 different cases to show that this was a relatively general thing. It's not always perfect, but it's a good way of estimating. But we can get resolution that matches pretty closely. So if you get a 26 angstrom resolution from comparing the two halves of your reconstructions, that tells you that it's probably about 26 angstrom resolution if you were to compare it to the actual known structure. I ran this with a lot of different experimental data sets. These were from that image I showed you earlier today, just using electron density. And the key is that this is now actual electron density functions. We've actually gone and solved the phase problem for solution scattering. And you can see these gradients of electron density in these profiles. Now, one thing to keep in mind is that these are low resolution electron densities. So if you actually think about what does low resolution electron density look like for perfect, perfect electron density, and that's what this is. If you actually calculate the electron density at low resolution from the model, you can see that in most cases it generally agrees. I mean, again, it's not perfect. It's SAXS, so it's low resolution, but for the most part, not only do the shapes, but the densities themselves agree. And again, you can kind of see that just like we saw from bead modeling, extra regions that were where the unresolved density from the crystallographic structure is corresponding to, say, 15 or 20% of the structure, you can actually see that in the density as well. And in this case, for example, you can see potentially why it's not resolved in the structure. It's less dense there, which means it's less rigid there, which means it's probably flexible, and so it's flopping around. This is just a comparison with the bead models. All right, so this is advantageous, not only because you're getting actual electron density, but for a few cases, there are, real useful reasons why we could use electron density. And one of those is when you have particles that have mixed species, okay? Good examples of that where you have something like protein and lipid, okay? Where lipid is very low density and protein is relatively high density. And so in this particular case, I found some experimental data, and this is available on the public SAS biological database. And so it was a really nice case because not only is it sort of this dumbbell shape, it's a very relatively complex shape for SAXS to reconstruct. It's not just a blob that you can fit anything to. It's a relatively well-defined shape that is pretty unique. And what happens is that these spheres on either end are lipid micelles. And so the green region is a protein dimer that's connecting the two micelles, and the cyan is spheres containing electron-rich head groups as the outer shell of the micelle, and low-density lipid tails as the inner shell. And so what this should show us is actually 3 different electron density regions. One for the high-density head groups, one for the low-density inner core region, and one for the protein. Just for reference, this was the ab initio bead model that was published along with it. You can see that, for the complex shape that it is, that this object is, it's actually pretty good. The overall shape is correct. You get blobs, maybe some evidence of some internal cavity, but you can see they're not spherical. They're, it's a good reconstruction, but we can do better. And so this is what we get from the electron density function. You can see that not only do we get nice spherical lobes, they're hollow shells so that we can actually see, this is just a cross-section of this up here. We can actually see the internal density fluctuations. We have a high density outer shell and a low density core on the inside, and we have our protein bridge connecting them. And so you can see that this was reconstructed to about 30 angstrom resolution. We can also look at the fact that we are actually reconstructing real density. That was one of the big questions about this. And so this was just a simple test that I ran to say that we are reconstructing actual density. And so I simulated some very simple spherical models. Okay? So here we have just flat electron density. This is a simulated reconstruction on the left. Not reconstruction, it's just a simulated density on the left, where I set all the density to the same number. Here I have a gradient, low density in the center to high density at the edge. Here I have the opposite gradient, where it's high density in the center to low density at the edge. And here, to make something that's not spherically symmetric, I just have two different uniform density regions that are offset by some distance from the center. And you can see that in the case of the flat electron density, it reconstructs the flat electron density for both single reconstructions and the average. And it does equivalently as what bead modeling does. Bead modeling is designed for exactly this thing. Uniform density, and spheres are like the ideal shape. So you can see that the single reconstruction and the average reflect the initial, low density in the center to high density at the edge, gradient. You can see that the bead model sort of tries to reconstruct that, but again, this is just damn if. We tried Monza, maybe it would do better, but you can see that it sort of tries to reconstruct the low density center with these cavities forming the inside. If you have high density in the center, which is sort of these inverse micelles, these reverse micelles that Bouchard was just talking about, we can see high density in the center and low density at the edge, and we effectively just see a sphere with the uniform density bead models. And probably the worst case is this sort of offset from the center, but you see that we kind of get a uniform density region here and a somewhat uniform density region here. It's not a perfect sphere, but it is offset by about the amount we expect. And you can see the corresponding bead model. So you can see that we're actually getting the actual density gradients that we would expect. So I went and simulated some complex shapes, okay, to see what are the limits, okay, because SAXS itself is still going to fundamentally be limited by the amount of information in the scattering profile. And in some cases it's pretty good, and in other cases it's not so good, okay? So in this case in particular, this is a really, really weird looking snowflake that you can see. It's sort of this very, very flat, sort of almost disc-like profile that's got these, immunoglobulin chains that are these sort of Y chains sticking out from the center. This is a very difficult thing to reconstruct, and my algorithm didn't do a fantastic job. The reconstructed resolution was something like 100 angstrom resolution, whereas this thing is like 350 angstroms across, right? And so this is a very high axial ratio, and it's got this very, intricate structure, very difficult. The overall size, it's got a disc-like profile, the overall size, it's got some lobes, but it's only reconstructing 2 of the 5 lobes in the center region. So it didn't do too great. So this is sort of an idea of what are the limits of the thing. It still, in my opinion, did a better job than bead modeling. Here's a nice example where bead modeling did a very good job reconstructing the shape. You can see, this is P1. You can see the, somewhat trimeric symmetry. But the added information that we get from the density profile, the density modeling, is that we can actually see these inner sort of low density core regions in these cavities. And here's just another example where we have a somewhat complex shape that bead modeling didn't do too well with, but we're able to reconstruct pretty well with the density. All right, so this is where this is at now. I'm working on a whole bunch of things to make this useful for everybody, including incorporating known structures. If you happen to know crystallographic symmetry, or rather non-crystallographic symmetry, can you apply P2, P4, P3 for known oligomeric states? Some density modification things to try and improve the resolution. This is really just a first pass, and it seems to be doing pretty well. I've got a version of this now. It's not public, but I'm working on publishing it, that allows us to work with mixtures and ensembles. So it would be a new alternative to the idea of sort of ensemble modeling. That seems to be working pretty well. I'd really like to see if we can actually use these electron density models as initial molecular replacement templates for crystallography. A neat thing is that because we're just using Fourier transforms, there's no assumption of atomic form factors. There's no form factors at all. It's not coarse-grained, it's not atomic, it's nothing. It's just Fourier transforms, which means it's directly applicable to effectively any type of scattering, including neutron scattering. And so I'm working on some versions that you could incorporate multiple different contracts and simultaneously fit all of these. And finally, some material science applications are possible too. So there's a whole host of things that I'm working on. All this code is open source in Python. You can find it at GitHub. I'm effectively the only person working on this, so if you want to take a stab at it, I'm more than happy to help you. It's called DENSE, which stands for Density from Solution Scattering. There's lots of info at this website. It's supposed to be dense.org, but I screwed up something with the domain name, so just go here. Dense.org probably will work, but depending on what system you're on. So really neat is Jesse integrated DENSE directly into RAW, and so you don't actually need to install DENSE to get the reconstructions. The problem is that the averaging isn't built into RAW. It's using a program called eman2, still relatively straightforward to install, but it's not something we can directly integrate into RAW yet. Also, if you just want to get a quick test of it, we have a server that we set up at UB called DenseWeb, and so you can run simple reconstructions. So I've set up DENSE to be as easy to use as possible, and so if you run defaults in DENSE, you can get pretty much publishable reconstructions. It sort of operates in slow mode, and you get sort of 20 reconstructions, and it averages the 20 reconstructions, and you can do like the enantiomer or filtering and things like that. And so you can get, using pretty much the defaults, you can get publishable reconstructions. Like I said, feel free to email me with any questions. I'm happy to, this is a very new thing. I'm constantly fixing bugs and things like that, so let me know if you find things. And this is a quick screenshot of the website. Go here, dense.org, and you can get information about how to install it and download it. This tutorial page, there's also a tips page if you hover over the tutorial thing. There's tons of information on how to actually go through it. It's really well described. And there's also the DENSE web server. You actually get this nice little interactive 3D representation of the density as well. Okay, so I can take any questions. That's it.
How do you decide the weight factor for the connectivity restraint for the cases where you have low density bridges connecting?
Yeah, so it's really stupid. Effectively, all it does is it looks at all the different blobs, and it says, find the blob that has the most density and get rid of all the other ones, and then just keep going. And so what'll happen is, most of the time it'll actually converge to one blob by the time you run this sort of connectivity restraint. But what'll happen is, it'll sort of be a shock to the system when you remove all these things, but it'll eventually get back to where it fits. And it does a pretty good job.
So like one example where you had this low density lipid tail within the protein densities. So did you have any problem assigning the connectivity restraint there?
Yep. So that was actually mainly the one case that I had to develop this connectivity thing for. For most simple globular things, it'll generally converge to a single blob. For that one, it had like 12. And so that was when I told it to look for all the, what would happen is there's these blobs, and then there's blobs really far away. And so what happens is, even though you would think that these really disconnected blobs would have a big impact, what happens is because they're so far away, the scattering vectors are extraordinarily small Q values, and so it doesn't really impact the fit all that much. But each one of the blobs will often look like this dumbbell thing. And so by deleting all the other dumbbells and just leaving one central one, it does a pretty good job of being able to reconstruct. That was a big part of making it work well.
Okay. And when you apply the mask for solvent patterning, is it a rigid mask or a soft mask?
It's a rigid mask. Yeah.
Rigid mask.
Yeah, so it's effectively what it does is it looks for, so it does a Gaussian blur. So if you think of like in Photoshop, you can just click blur Gaussian, and it'll take your 2D image and get rid of all the noisy stuff and just blur it, just like you blur a camera lens. And then what it does is it says, okay, what's the maximum intensity? And it says take 20% as your threshold. And so these are all editable parameters. You can change the threshold, change all these things. So you set your threshold to be 20%. Everything below 20% in the blurred map will get told to be solvent. That's sort of outside the support. And then everything above 20% is inside the support. And it does this gradually. So when it starts, for the first, like, I don't know, 5,000 steps or something, it's not using any support at all. There's no solvent flattening. It's just positivity. And that gets really pretty close to what the final answer should be. And then the solvent flattening gets engaged, and it just kind of cleans it up. So the solvent flattening at this stage is really just cleaning up the noise.
So even we do buffer subtraction when we collect the data, we have to do solvent flattening.
Yeah, sure. So in this, for now. So one of the versions I'm working on, I have like 37 different versions of this thing with all different, all my different branches. One of the versions I'm working on could potentially eliminate the need for buffer subtraction, or at least get around to potentially getting correct buffer subtraction. And so it does this by actually modeling what the buffer looks like. And actually, like, I've run this, there's no necessarily restraint that it has to be protein. So I've actually run it with just water, and it actually reconstructs the density of water pretty well. And there's some things you have to do to allow it to do that, like turning off solvent flattening, because it's solvent flat, the solvent's not flat anymore. But there's a version I'm working on that kind of combines these two ideas so that you can actually model the solvent and model the protein simultaneously. And this is the version that sort of does this contrast matching with neutron data, because you have the same molecule, but different levels of buffer contrast. So that's the basic idea.
One last question. It's a very general question, like what limits the resolution in a SAXS experiment? Like one of the density reconstruction model did, like the one which had the best resolution was 16 angstrom. That protein is very small, it has floppy regions, but it has the best resolution. The bulkier ones are like 28 angstrom.
Yeah, so it's a combination of size and complexity. So something like this is very globular and it's very small. So I think the whole distance here is something like 50 angstroms across, right? So if you think about it, a 16 angstrom resolution, you're talking 3 or 4 pixels, right? You're really only seeing 3 or 4 pixels worth of information. So it's not actually that surprising that it's that high, but something here, this is 100 angstroms across. So again, you're looking at something like, 3 or 4 pixels across, right? But something that's really complex, like something that's elongated, tends to have a lower resolution than something that's globular. And that's just because, sort of this idea of ambimeter, there's just implicitly more complexity to the shape of certain objects, like just the shapes. If you've got some crazy, like that snowflake looking thing, that's a really complex shape in addition to being a large and flat object. So that's sort of, it's a combination of the size of the object and the complexity of the shape. Sounds good.
All right, so that concludes the lectures for today. So tomorrow morning there are software tutorials in this room. I believe it starts at 8:30. There will be food probably very similar to what there was this morning, so calibrate your breakfast needs accordingly. And some of you are collecting data tonight. Some of you will be tomorrow or Sunday. And other than that, besides data collection and lectures, there's nothing else to do, so enjoy Ithaca. You have the evening off. And we'll see you all here tomorrow morning. If you have any questions related to course stuff that you need to ask now, come catch me.